Amphibian populations are declining worldwide for many reasons. One reason is the contamination of wetland habitats by pesticides. Pesticides are easily absorbed by amphibians and can negatively affect their health. Interestingly, even levels considered safe have sublethal effects on amphibian biology. Exposure to trace amounts of Chlorpyrifos, a common pesticide applied to fruits and vegetables, resulted in altered brain shape and elevated glucocorticoid hormones in tadpoles. Glucocorticoids are hormones involved in the metabolism of carbohydrates, proteins, and fats. The most prominent glucocorticoid in amphibians is corticosterone. While the release of glucocorticoids is beneficial in short term, prolonged increase in glucocorticoids can be detrimental. A previous study tested the effects of CPF in court on development by exposing northern leopard frog tadpoles to CPF court or a vehicle control. Here, I tested whether these treatments affected liver morphology and the immune system response. Sublethal pesticides cause liver damage both morphologically and histologically. I predict that tadpoles exposed during development to CPF or court will have altered liver morphology, and that tadpoles exposed during development to CPF or court will have decreased melanomacrophage content relative to controlled tadpole groups. Melanomacrophages are a type of immune cell found in livers that have a dark appearance as they contain melanin. I used 30 tadpoles, Lithobates pipiens, per treatment that were exposed for 7 weeks, starting at hatching. I then dissected the tadpoles and stained the liver tissue. The liver and pancreas were removed and weighed. To correct for body mass, organ masses were corrected for differences in body mass. To determine basic morphology, I stained with H&E, and slides were stained with PAS to look for concentration of glycogen in liver tissue. To assess for depleted immune system, the number of melanomacrophages were counted and compared to the control group using image J. My results supported our hypothesis that CORT mediates liver growth during development. Specifically, CORT decreased relative liver and pancreas mass. Unlike my hypothesis, CPF did not affect liver mass. Tadpoles treated with CPF remain similar to the vehicle control. The decrease in liver mass suggests that CORT, as a metabolic hormone, is breaking down liver glycogen stores. The average density of melanomacrophages in treatment group was compared. From this data, the CORT treatment group shows a decreased trend in melanomacrophage density. This helps support the hypothesis that stress impacts the immune system. However, more research is needed. Future directions would include analyzing how CORT and CPF affect glycogen concentration in the liver, and with my observations in melanomacrophage, this research can be used to analyze how stress impacts the liver in a larger study. Overall, CORT mediates liver growth during development. The reduction of liver mass by stressors that cause an increase in CORT may affect organismal performance and fitness. Finally, I'd like to thank the Woodley Lab and the Department of Biological Sciences for their support. I also thank Tri Beta for funding this research.